Okay, welcome everyone. I am following in the path of Malike Lakadamiali, who's talking about mechano-intelligence and how it relates to a specific part of the nucleus cell tissue spectrum. My charge in this talk is to talk to you about mechano-intelligence specifically as it relates to the dialogue between a cell and its matrix, or in the case of plants, the cell wall, and to review recent advances in the field and also to highlight where the field needs to go. So I want to introduce the term recursive dialogue to you. And this is a term that's highly relevant to thinking about cells and their ECM or cells in their cell wall. And it's a term that suggests that cells in the matrix change each other over time, that the nature of the communication changes the communicators, which in turn communicate differently. So the essential questions that are raised by this concept and that we think about a lot in CEMB are: how do cells respond to and discriminate between different kinds of mechanical stimuli? How do they integrate internal and external mechanical and biochemical signals? And that's important. I think Malike touched on that a little bit about how, you integrate signals from the nucleus and the extracellular matrix. And finally, the question of mechano-intelligence, which is that can we engineer cells and ECMs and cells and cell walls to create desirable outputs? And so, in other words, can we engineer cells and their environments to make them able to, when they need to, and only when they need to, respond to and change each other in beneficial ways? So, for example, to produce an altered matrix environment, to heal a wound in a regenerative rather than reparative or scarring fashion, or to alter cell migratory capabilities, but only in the context of malignancy or pre-malignancy. In plants, this might be translated as asking whether we can engineer the cell wall to respond in protective ways in the setting of drought, but not when there's a lot of rain, or how to fight off invaders, but not to set into effect those pathways if there aren't invaders. So the cartoon here gives you some concept of this, some idea of this concept of recursive dialogue. And it gives you a sense of why it falls under the heading of mechano-intelligence, I think. And it's cells in their environment, if you look on the left, can be assumed to have a relatively stable set of interactions in the context of homeostasis, as I've shown there. There are loops, and they really are essentially unchanged loops. But the second cartoon on the right suggests that in the context of some kind of threat, either disease or some other kind of perturbation, there's a need to respond. And what might happen is that a cell communicates with its matrix, and that it changes the matrix, and that the matrix in turn, the changed matrix in turn, causes the cell to change, and the changed cell changes the matrix further, and so on until you end up with a cell and a matrix that are both changed in a way that responds to the threat or the perturbation. So it's really a reiterative back and forth between cells and the environment, and that's really how we think of recursive dialogue. So my goal in this talk is to show you that mechanobiology, particularly in the last decade, has made incredible strides towards the goal of engineering mechano-intelligence, particularly as far as cell matrix interactions go. And this is in part, I think, because of a new appreciation for time and length scales in cell matrix interactions. And I hope this will become clear as I continue to talk. So what I'm going to cover in this brief talk is give you a sense of the varied mechanical stimuli that impact animal cells, and then talk briefly about mechanics in 4D in the plant cell wall that I hope at that point you'll be able to appreciate is very similar to what I've told you about mechanical stimuli in animal cells. And then we'll talk a little bit about time and length scales and how they serve as a link between cell, nuclear, and tissue mechanics. And finally, I'll talk in a little bit more depth about tissue mechanics and about how what we've talked about as far as cell matrix and cell cell wall interactions leads to the need for and the potential for new materials and models. So I think, as everyone in this audience knows, one of the most important milestones in mechanobiology was this 1997 paper from Pelham and Wang that demonstrated that cells on variable stiffness gels had altered cell behavior depending on what that stiffness. And they particularly showed that this was the case with altered migration. So since then, this has led to many, many, many papers exploiting the method. Probably most of us here have carried out experiments with polyacrylamide substrates of various stiffness. CEMB members Dennis Disher and Chris Chen have done some particularly notable work with this system. And so there have now been really extraordinary advances using this system in understanding how cells interact with their environment and appreciating that differentiation, migration, growth, in some cases malignant transformation, and a host of other behaviors are impacted by environmental stiffness. But I also want to emphasize that although it continues to be very widely used, there are significant limitations with the use of polyacrylamide as it has been used. And part of that is that polyacrylamide is a linearly elastic material. And it varies, it has a very limited variation in time and length scales. And this is not what happens in the case of biomaterials. So biomaterials demonstrate a variety of different mechanical properties that do involve time and length scales. And those include, as shown here, viscoelasticity, viscoplasticity, poroelasticity, other kinds of nonlinear elasticity. Generally, biomaterials, most of them are fibrous networks. And this is all different than what a standard polyacrylamide gel is. And that these different properties can impact proliferation and apoptosis, differentiation, migration, metabolic behavior, among others. And that at a broad scale, this influences development, homeostasis, wound healing and regeneration, and sort of a response to injury. And that these are all really critical to think about as we look at this concept of a recursive dialogue between cells and their environment. And when we ask cells in the environment to start responding in sort of on-demand ways that are beneficial to the particular problem that a cell or the environment is facing at any given time. So the first, what I want to do is I want to go through a variety of examples of how biological materials have influenced the development of synthetic materials that have some of these properties, and how that has led to really new insight into the impact of time and length scales on cell behavior. I'm going to review these, speaking about work done primarily by people in CEMB, and these are phenomenal examples of this kind of work. But I certainly have to acknowledge that this kind of work has been carried out by a number of fantastic labs outside of CEMB, and I certainly acknowledge their contributions as well. So the first one I want to talk about is viscous dissipation. And this is work from Paul Janmay's lab, and specifically a postdoc named Elizabeth Charrier. And she considered the fact that tissues act as viscoelastic solids, where they typically have loss moduli of 10 to 20% of the value of the elastic modulus. And so what Elizabeth did was she developed new materials that were composed of cross-linked polyacrylamide, which is not showing up over there because it's too light. There's light gray squiggles over here, cross-linked polyacrylamide, and linear polyacrylamide, which is viscous, and that the combination gives you a viscoelastic gel. And the key point about these materials is they have independently tunable elastic and viscous moduli. And so what Elizabeth did was she used these gels and looked at what happened to behavior when she held the elastic modulus constant, but she changed the viscous modulus. And if you look at the bottom here, these are MSCs, I believe, although it's also been seen with a primary cell type from the liver, where she held G prime at 5 kilopascals, but she varied G double prime from 0 to 500 pascals. And she examined these cells over 7 days. And what she found was that when the G double prime was at 0 or at 200, there were marked changes in cell area over 7 days. But when she increased the G double prime to 500, she did not see that impact. There was no change. And so the conclusion from this, and obviously I've summarized her work, is that viscous dissipation influences cell phenotype and behavior, in this case, including differentiation. That the work was greatly expanded then by a collaboration between the Jan May lab and Vivek Shenoy's lab, developing modeling to explain what was happening and to define the impact of viscosity in a bit more detail. And so what the Shenoy lab did was develop a motor clutch model that simulated focal adhesions, and that's shown up here at the top. And what was found in these simulations, as shown at the bottom, is that intermediate viscosity maximizes cell spreading on soft substrates, but that it was independent of viscosity on stiff substrates. And the model enabled them to reach the conclusion that viscosity leads to stiffening of soft substrates on a faster time scale than the clutch off rate. But on stiff substrates, the bound clutches were saturated. And so this was an interesting insight into the impact of viscous dissipation on cell behavior in the context of different stiffnesses, and it has been experimentally validated. So I just want to highlight that this demonstrates a really critical relevance of time scales, and of in this case, of relatively fast time scales of viscous dissipation, but the importance of time scales in the environment in determining cell behavior. So the second finding I want to talk about is an earlier finding, actually, again from the Shenoy group, that is related to matrix mechanics. This is a finding that I think continues to resonate, and you'll see that it comes back through this talk, showing the role of fibrous networks and of tension-driven alignment of fibers in long-range force transmission. So if you look at the two panels on the left, you can see first on the far left a simulation of a random network. So these are cross-linked but randomly aligned fibers. When a stress is applied, you can see, or tension is applied, you can see actually that some of the fibers align, although some buckle. But those that align are able to transmit force, as you can imagine, over lengths much greater than what the diameter of a cell might be, and much greater than the length of the individual fiber. And this is demonstrated in these figures on the right-hand side, where the simulation is of a cell in different kinds of matrices. And the question is, what is the distance of the force transmission through that substrate? So you can see that with the linear substrate and two different kinds of neo-Hookean substrates, the transmission is not particularly great compared to the diameter of a cell. But when you put the cell in a fibrous substrate, in fact, transmission can be much, much greater, potentially 10 times or more the length of the diameter of a cell. So this is an important mechanism of long-range force transmission. It requires that the cell be in a fibrous network, in other words, cross-linked fibers, and it demonstrates the relevance of length scales. This work was actually expanded in an important way, actually about 8 years after the original paper, with the demonstration that the addition of other matrix components to a fibrous network could significantly impact the length of long-range force transmission. So in this case, glycosaminoglycans, and it's specifically hyaluronic acid for people who are here at the last talk, when when added to the fibrous network, cause a significant decrease in force transmission distance. And what this suggested is, I mean, first, obviously that glycosaminoglycans can alter the length scales of cell mechanical signaling, but it was also demonstrated that variable mechanics is really key in variable matrix can be really quite key in determining some of the complex mechanics of the tissue. And because cells synthesize components like hyaluronic acid, I mean, in addition to collagen, obviously, that cells can regulate matrix mechanical signaling via not just contractility, but also through synthesis and deposition of other matrix components. A third aspect that I wanted to discuss as far as considering how matrices are more complicated than linearly elastic polyacrylamide is the concept of plasticity. And that is where there is the persistence of changes even after removing whatever the tension or the force is. So in this case, this is work with Vivek Shenoy and also my group, but primarily out of Avi Chowdhury's, actually with a collaboration of Avi Chowdhury at Stanford. You can see in the upper left that two cell clusters are in collagen, and that because of their contractility, they're actually able to pull on the fibrous network and align that collagen, just as I'd shown you in the simulation in the previous slide. But the interesting thing is that if you treat these cells after this alignment has occurred with blebistatin, or you treat with to remove contractility, or you treat with trypsin to completely remove cells, the majority of that alignment and the collagen reorganization persists. And this is quantified here on the lower left. So, so in other words, the matrix is plastic. You can apply a force to it, and that the changes are permanent, at least some of them. And the Shenoy lab did some theory and simulations to explain the mechanism of this. And it appears to be primarily the breakage of some bonds and then the formation of new weak cross links after network stretching. And there did seem to be some component of permanent fiber elongation and sliding. Another paper from the Shenoy lab, as well as the Chowdhury lab, looked at cancer cell invadipodia, and this is from a couple of years ago, and were able to show that viscous dissipation was able to soften the ECM over repeated cycles, and that as a result, there was accumulated plastic deformation due to the cyclic ratcheting. And you recall the model of viscous dissipation that I showed you a couple slides ago. And so this was critical work in that it highlighted the essential combination of different time and length scales in biological matrices and in determining the behavior of cells in that matrix. So in this case, cancer cell invadipodia depended on these invadipodia changing the ECM leading to changed invadipodia, and so on and so on, until there was accumulation of really quite significant deformation. So I told you a lot about the animal cell matrix. How does this relate to plants? Because we are a center that thinks about both plants and animals. And so I want to mention to you this series of really beautiful papers from Dan Cosgrove's lab at Penn State that showed that the cell wall, in this case the onion epidermis, but I think it's likely to be quite generalizable, is mechanically complex. And I just want you to note that when I'm telling you about the plant cell wall from the Cosgrove Lab's work is quite similar to what you've heard about the animal cell ECM. So they were able to show that the plant cell wall is highly nonlinear in its stress strain behavior. And that interestingly, it behaved like a network of cellulose fibers rather than a viscoelastic pectin-dominated material. There was plasticity from fibril sliding with a small component of permanent microfibril straightening in cellulose networks that were aligned, and that the timescales were really critical to these events. And so in addition to the fact that this is very similar to much of what we learned about the animal ECM, it suggests that understanding these mechanics of the plant cell wall, like understanding the ECM related to animal cells, are going to be critical for the regulation of morphogenesis and structure. Although the first talk today was on somewhat of a different topic, I think you got, for people who are not in the plant world, you got a sense of how plant cells, their shape and their size are really very much determined by their plant cell wall. And so these findings are really critical to sort of the structure of plant cells and the tissue and of morphogenesis. They specifically looked at the impact of drought, in other words, dehydration, and were able to show that the cellulose network sliding and extensibility were really critical it caused significant changes in the mechanics of the cell wall. But this, again, is an important point to consider in thinking about how these mechanical properties of the cell wall may have an important impact on the response to a perturbation. Although it has not yet been done, to my knowledge, these findings are highly applicable to plant tissue mechanics. So I want to emphasize what I've told you, that both plant cell walls and the animal ECM demonstrate features like plasticity, viscoelasticity. They have fibrous network behaviors and other complex mechanics that go beyond just linear elasticity. And that time and length scales, sort of the fourth dimension, are particularly important. And that cells respond in very profound and sort of basic ways with altered growth and differentiation, proliferation, and migration. And I want you to think back to the talk that Malike Lakadamiali gave this morning and think about the fact that cell ECM interactions are driven in part by what cells do, namely contractility, and by what they make, what they synthesize. And much of that is actually driven by events occurring within the nucleus. And so you cannot separate out nuclear mechanics and cell ECM mechanics from a consideration of nuclear mechanics. They're all quite connected. And so I just want to remind you again of this concept of mechano-intelligence and this idea of recursive dialogue, and specifically the need to respond when there's a perturbation. So one of the key questions then is, how do we develop materials that are going to help us study this, and mechano-intelligent materials and materials that we could actually use for plant biology or for animal biology? And there are a lot of very exciting and new mechanically responsive materials that are available. And I want to highlight this one in particular. It's from Matt Davidson and Jason Burdick's group. And it's important because it's actually a fibrous material that's mechano-responsive. And so what Matt did was carried out electrospinning, and this is norbornene HA, carried out electrospinning to generate fibrous hydrogel networks, and then modified distinct fiber populations with complementary chemistries. In one case, one of either the red or the green is modified with hydrazide, and the other is modified with aldehydes. And under mechanical loading, when those two kinds of fibers get close together, chemical reactions occur, and those chemical reactions can be permanent. And so what Matt was able to show was that in this system, when he caused deformations, those deformations were permanent. And I just want to say 1, 2, and 3 correlate with before, during, and after a deformation or the application of a stress or tension. And you can see that in both the top row, you can see that there's that deformation. And even with removal of the force, you can see that it's a plastic or permanent deformation. In the synthetic matrix below, which is the same matrix, you can see that with the application of additional tensile stress, you can see the same thing, that there are permanent changes. In collaboration with Vivek Shenoy's group, they worked on some theory and were able to show that these deformations, and particularly their permanence, was completely predictable. And that's important if we consider how these might be used in the future. So this is an important example of a fibrous network that can adapt to loading. And I think there will be likely many more to come. Now, I did want us to think a little bit just about the new materials that are needed for engineering mechano-intelligence. And this is actually a figure from Matt Davidson. And I just wanted to highlight that he's in this cartoon talking about new materials needed. He's highlighting that we need materials with time responsiveness over a variety of length scales. And I just want to, in a little bit more detail, go into some of the specific features of synthetic materials that would be required to generate these synthetic materials. So in the case of nonlinear elasticity, we need reorientation of load-bearing elements. For viscoelasticity, it would require potentially breaking weak crosslinks, releasing entanglements, or unfolding proteins, and so on. And so there are many potential materials out there, and obviously many ways to get them, and many labs working on this. So finally, I just want to talk about tissue mechanics. So I've talked a lot about the cell matrix dialogue, and I've mentioned how this relates to nuclear mechanics and the mechano-intelligence that Malike Lakadamiali discussed. But I want to talk briefly about tissue mechanics and how this cell matrix dialogue can help resolve this paradox of animal tissue mechanics. And what that paradox is, and it's something that Paul Janmay has highlighted, is that collagen, as shown here, softens with axial strain. And yet tissues, which consist primarily basically of cells within collagen, stiffen with axial strain. And how do you explain that? And so Anne Van Oosten, who is a grad student in Paul's lab, in collaboration with Paul and with Vivek Shenoy's group, have developed, first demonstrated this very nice, this is a tissue mimic system where she added various volumes of particles to a fibrin-fibrous network, and was able to show that once she reached a threshold of particles, she was able to generate compression stiffening. And so Vivek's lab developed a model which was able to take the concept of incompressible cells in a matrix with the properties that we've discussed, namely collagen that can compression soften, collagen that is fibrous, and where you can have alignment of some fibers and buckling of other fibers, and use that to explain exactly how you could have cells within a collagenous matrix, and yet nonetheless you'd get compression stiffening. So this is one way to envision sort of tissue mechanics and how it's related to this recursive dialogue between the cell and the matrix. So looking ahead at this concept of a recursive dialogue between cell and matrix, we need to develop mechano-intelligence for mechanical and agricultural applications. And there are really incredible possibilities. I've mentioned many of them: wound healing, sort of the regulation of cell roots, of how plant cells respond to drought. This is inextricably linked to tissue and nuclear mechanics, and what cells make, the forces they generate, and how that influences their wider tissue environment. We do need new tissue models that incorporate both plants and also the many kinds of mechanics that we see in animal biomaterials. And we do need new synthetic and quasi-synthetic materials and approaches to make this happen. So I want to finish up here and just say, if you have any questions or comments, and these talks are intended effectively to lead to a recursive dialogue between attendees at the conference. So either now or later, I think that would be a great thing to have happen. Thanks.
Thank you, Dr. Wells, for that awesome talk. We have time for 1 or 2 questions.
I teach a class with someone, and the deal is that if you just stare at them long enough in silence, someone will talk. And the question is, who's going to cave? So In our class, we know exactly who it is, but.
Thank you, Becky. That was really a great overview. I just was wondering your thoughts in your work. You know, you've talked a lot about time scales, length scales as it relates to the ECM. And then, you know, there's been a lot of work at the center about time and length scales intracellularly.
Yeah.
I wonder if we also thought about in terms of multicellular, like organ level time and length scales, and how like a millimeter or 100 micron, you know, body may interact with ECM differently than single cells or subcellular length.
Yeah, I mean, you're talking about tissue mechanics?
Yeah, like.
Yeah, so I think Trina Arinze is going to talk more about that, and but it is an interesting question. How, you know, is there anything different when you start at making it complex and multicellular? Does it follow completely? Does the theory actually cover everything we see? I don't know that. And your other point about the cytoskeleton was a good one. I think we tend to think of, Vivek, correct me if I'm wrong, I think we tend to think of the cytoskeleton as being very similar to matrix and its impacts. And we have some of the data to suggest that's the case. Yeah, so.